Hello friends, my seminar topic is nucleus, structure and function of nucleus. Introduction. Nucleus is the brain of the cell. It is a membrane-bound organelle that contains genetic material, DNA, of eukaryotic organisms. As such, it serves to maintain the integrity of the cell by facilitating transcription and replication process. Robert Brown first reported nucleus in 1831. It's the largest organelle inside the cell, taking up about a tenth of the entire cell volume. This makes it one of the easiest organelles to identify under the microscope. Next one, structure and organization of the nucleus. As the organelle that contains of genetic material of a cell, the nucleus can be described as the command center of a cell. As such, the nucleus consists of a number of structured elements that allow it to perform its functions. In general, nucleus has a spherical shape. However, it may appear flattened, ellipsoidal, or irregular depending on the type of cell. For instance, the nucleus of columnar epithelial cells appears more elongated compared to those of other cells. The shape of nucleus, however, may also change as the cell matures. Structure of nucleus encompasses the nuclear membrane, nucleoplasm, chromosomes and nucleolus. Next one is the nuclear envelope or membrane. Considering nucleus, its importance in the storage and utilization of genetic information, the nucleus of a eukaryotic cell has at first glance a rather undistinguished morphology. The contents of the nucleus are present as a viscous amorphous mass of material enclosed by a complete complex nuclear envelope that forms a boundary between the nucleus and cytoplasm. First one, the chromosomes, which are present as highly extended nucleoprotein fibers termed chromatin. Second one or more nucleoli, which are irregularly shaped electron-dense structures that function in the synthesis of ribosomal RNA and the assembling of ribosomes. And the third one, the nucleoplasm, the fluid substance in which the solutes of nucleus are dissolved. The separation of a cell's genetic material from the surrounding cytoplasm may be the single most important feature that distinguishes eukaryotes from prokaryotes, which makes the appearance of the nuclear envelope a landmark in biological evolution. The nuclear envelope consists of two cellular membranes arranged parallel to one another and separated by 10 to 15 nanometer. Together, these membranes contain upward of 16 distinct membrane transmembrane proteins, including a number of species that link the outer nuclear membrane with elements of the cytoskeleton. The inner and outer nuclear membranes are fused at sides forming circular pores that contain complex assemblies of proteins. The average mammalian cell contains several thousand nuclear pores. The outer membrane is generally studded with ribosomes and is continuous with the membrane of the rough endoplasmic reticulum. The space between the membranes is continuous with the endoplasmic reticulum lumen. The inner surface of the nuclear envelope of animal cell is bound by integral membrane proteins to a thin filamentous meshwork called the nuclear lamina. The nuclear lamina provides mechanical support to the nuclear envelope, serves as a site of attachment for chromatin fibers at the nuclear periphery, and has a poorly understood role in DNA replication and transcription. The filaments of the nuclear lamina are approximately 10 nanometer in diameter and composed of polypeptides called lamins. Lamins are members of the same superfamily of polypeptides that assemble into the 10 nanometer intermediate filaments of the cytoplasm. The disassembly of the nuclear lamina prior to mitosis is induced by phosphorylation of the lamins. Mutations in one of the lamin genes are responsible for a number of diverse human diseases, including a rare form of muscular dystrophy called EDMD2, in which muscle cells contain exceptionally fragile nuclei. Mutations in lamins have also been linked to a disease called Hutchinson-Gilford progeria syndrome (HGPS) that is characterized by premature aging and death during teenage years from heart attack or stroke. Next one is the nuclear pore complex and its role in nuclear cytoplasmic trafficking. The nuclear envelope is the barrier between the nucleus and cytoplasm, and nuclear pores are the gateways across that barrier. Unlike the plasma membrane, which prevents passage of macromolecules between the cytoplasm and the extracellular space, the nuclear envelope is a hub of activity for the movement of mRNAs and proteins in both directions between the nucleus and cytoplasm. The replication and transcription of genetic material within the nucleus require the participation of large numbers of proteins that are synthesized in the cytoplasm and transported across the nuclear envelope. Conversely, mRNAs, tRNAs, and ribosomal subunits that are manufactured in the nucleus must be transported through the nuclear envelope in the opposite direction. Some components, such as the snRNAs of the spliceosome, move in both directions. They are synthesized in the nucleus, assembled into RNP particles in the cytoplasm, and then shipped back to the nucleus where they function in mRNA processing. To appreciate the magnitude of the traffic in between the two major cellular compartments, consider a HeLa cell, which is estimated to contain about 10 lakh ribosomes. To support its growth, a single HeLa cell nucleus must import approximately 5,16,000 ribosomal proteins and export approximately 14,000 ribosomal subunits every minute. In one early approach, a suspension of tiny gold particles was injected into cells and passage of the material through the nuclear envelope was observed with the nuclear electron microscope. These particles move from the cytoplasm into the nucleus by passing single file through the center of the nuclear pores. Electron micrographs of cells fixed in the normal course of their activities have also shown that particulate material can pass through a nuclear pore. Materials are as large as gold particles and ribosomal subunits can penetrate nuclear pores. One might assume that these pores are merely open channels, but just opposite is true. Nuclear pores contain a doughnut-shaped structure called the nuclear pore complex (NPC) that straddles the nuclear envelope, projecting into both the cytoplasm and nucleoplasm. The NPC is a huge supramolecular complex, 15 to 13 items the mass of a ribosome that exhibits octagonal symmetry due to the eightfold repetition of a number structures. Despite their considerable size and complexity, NPC contains only about 13 different proteins called nucleoporins, which are largely conserved between yeast and vertebrates. Each nucleoporine is present in multiple copies, 8,
16 or 32 in number, in keeping with the octagonal symmetry of the structure. At the heart of the NPC is a central channel which is surrounded by a ring of nucleoporins whose rearrangements can change the diameter of the opening from about 20 to 40 nanometer. The NPC is not a static structure as evidenced by the finding that many of its component proteins are replaced with new copies over a time period of seconds to minutes. Recent studies suggest that this dynamic exchange of nucleoporins may play a role in activating the transcription of chromatin that is associated with the NPC. Among the nucleoporins is a subset of proteins that possess within their amino acid sequence a large number of phenylalanine glycine repeats. The phenylalanine glycine Fg repeats are clustered in a particular region of each molecule called the Fg domain. Because of their unusual amino acid composition, the Fg domain possesses a disordered structure that gives them an extended and flexible organization. The Fg repeat containing nucleoporins are thought to line the central channel of the NPC with a filamentous Fg domain extending into the heart of the central channel. The Fg domains from a hydrophobic meshwork or sieve that blocks the free diffusion of larger macromolecules between the nucleus and cytoplasm. In 1982, Robert Lasker and his co-workers at the Medical Research Council of England found that nucleoplasmin, one of the more abundant nuclear proteins of amphibian oocytes containing a stretch of amino acids near its C terminus that functions as a nuclear localization signals NLS. This sequence enables a protein to pass through the nuclear pores and enter the nucleus. The best studied or classical NLSs consist of one or two short stretches of positively charged amino acids. The T antigen encoded by the virus SV14, for example, contains an NLS identified as proline, lysine, 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 arginine, lysine, valine. If one of the basic amino acids in the sequence is replaced by a non-polar amino acid, the protein fails to localize to the nucleus. Conversely, if this NLS is fused to a mononuclear protein such as serum, albumin and injected into the cytoplasm, the modified protein becomes concentrated in the nucleus. Thus, targeting of proteins to the nucleus is similar in principle to trafficking of other proteins that are destined for segregation within a particular organelle, such as a mitochondrion or a peroxisome. In all of these cases, the proteins possess a specific address that is recognized by a specific receptor that mediates its transport into the organelle. The study of nuclear transport has been a very active area of research, driven by the development of in vitro systems capable of selectively importing proteins and RNPs into the nucleus. Using these systems, researchers have identified a family of proteins that function as a mobile transport receptors ferrying macromolecules across the nuclear envelope. Within this family, proteins have macromolecules into the nucleus from cytoplasm. and exportins move macromolecules in the opposite direction. Major steps that occur during the nuclear import of a protein, such as nucleoplasmin that contains a classical NLS. Import begins as the NLS containing cargo protein binds to a heterodimeric soluble NLS receptor called importin alpha or beta that resides in cytoplasm. The transport receptor is thought to escort the protein cargo to the outer surface of the nucleus where it likely docks with the cytoplasmic filaments that extend from outer ring of the NPC. A number of gold particles bound to those filaments. These particles were coated with an NLS containing nuclear protein that was being transported through the nuclear pore complex. The receptor cargo complex then moves through the nuclear pore by engaging in a series of successive interactions with the Fg domains of the Fg containing nucleoporins, allowing passage of the receptor cargo complex through the NPC. Once bound cargo proceeds through the NPC, a GTP binding protein called RAN drives release of the transported protein into the nuclear compartment. Like other GTP binding proteins such as SAR and EFTU, RAN can exist in an active GTP bound form or an inactive GDP bound form. RAN's role in regulating nucleocytoplasmic transport is based on a mechanism in which the cell maintains a high concentration of RAN-GTP in the nucleus and a very low concentration of RAN-GTP in the cytoplasm. The steep gradient of RAN-GTP across the nuclear envelope depends on the compartmentalization of certain accessory proteins. One of these accessory proteins, named RCCI, is sequestered in the nucleus where it promotes the conversion of RAN-GDP into RAN-GTP, thus maintaining the high nuclear level of RAN-GTP. Another accessory protein, RAN-GAPI, resides in the cytoplasm where it promotes the hydrolysis of RAN-GTP to RAN-GDP, thus maintaining the low cytoplasmic level of RAN-GTP. Thus, the energy released by the GTP hydrolysis is used to maintain the RAN-GTP gradient across the nuclear envelope. As discussed below, RAN-GTP gradient drives nuclear transport by a process that depends only on receptor-mediated diffusion on motor proteins or ATPase have been implicated. In classical NLS import pathway, when the importing cargo complex arrives in the nucleus, it is met by a new molecule of RANGTP which binds to the complex and causes its disassembly as indicated in step 4. This is the apparent function of the high level of RAN-GTP in the nucleus. It promotes the disassembly of complexes imported from the cytoplasm. The imported cargo is introduced into the nucleoplasm and one portion of the NLS receptor, the importin beta subunit, is shuttled back to the cytoplasm together with the bound RAN-GTP. Once in the cytoplasm, GTP molecule bound to RAN is hydrolyzed, releasing RAN-GDP from the importing beta subunit. RAN-GDP is returned to the nucleus where it is converted back to the GTP-bound state for additional rounds of activity. Importin alpha is transported back to the cytoplasm by one of the exportins. RAN-GTP plays a key role in the escort of macromolecules out of the nucleus just as it does in the import from the cytoplasm. Recall that RAN-GTP is essentially confined to the nucleus, whereas RAN-GTP induces the disassembly of imported complexes as shown in step 4. RAN-GTP promotes the assembly of export complexes. Proteins exported from the nucleus contain amino acid sequences called nuclear export signals or NESs that are recognized by transport receptors that carry them through the nuclear envelope to the cytoplasm.
Nuclear envelope remodeling in dividing cells. Although a member enclosed nuclear genome can be found in all eukaryotes, there is a critical difference in cell cycle dependent dynamics of nuclear envelope between lower eukaryotes, yeast, and metazoan. The former undergo closed mitosis, where spindle microtubules can either form inside the nucleus or are able to penetrate an intact nuclear membrane. In contrast, nuclear envelope of metazoan cells completely disintegrates during cell division to allow the mitotic spindle to access chromosomes. As a consequence, every divided cell has to reform the nuclear envelope and re-establishes the identity of the nuclear compartment. Nuclear envelope remodeling in proliferating cells is a highly dynamic process that involves a vast number of molecular players. By the end of interphase in G2, the nuclei have duplicated their genome, doubled the number of NPCs, and increased the surface area of nuclear envelope. The surrounding endoplasmic reticulum network is continuous with the nuclear envelope but not enriched in nuclear envelope proteins. The entry of mitosis, that is, prophase, is marked by nuclear envelope breakdown and the loss of the nucleocytoplasmic compartmentalization. Between nuclear envelope breakdown and early anaphase, when chromosomes align in the metaphase plate and subsequently segregate, chromatin is essentially free of membranes. During these cell cycle stages, the majority of soluble nuclear envelope proteins are distributed throughout the cytoplasm and transmembrane nuclear envelope proteins resides in the mitotic and endoplasmic reticulum. In anaphase, endoplasmic reticular membranes begin to reassociate with and rapidly enclose the chromatin mass. Chromatin association of a subset of NUPs, decondensation of chromatin, and the assembly of new NPCs occurs concomitantly with nuclear envelope formation. At the end of the cell division, the nuclear envelope has reformed as a closed membrane barrier and re-establishes the nuclear compartment by enabling selective nucleocytoplasmic transport. After its formation, the nuclear envelope expands and undergo additional structural changes necessary for cell cycle progression and transcription, including the assembly of new nuclear core complexes. Breaking down the nuclear envelope. Living cell imaging of mammalian cell has revealed that nuclear envelope breakdown, which occurs rapidly within a few minutes in prophase, occurs in a stepwise fashion. One of the initial events is the selective loss of NUPS from the NPCs. NUP98, a dynamic NPC component, is released before the rest of NPC component, which disassemble synchronously. Collective dispersal of NUPs can also be observed in Drosophila, suggesting that port disassembly is evolutionarily conserved. Protein kinase 1 may require access to proteins of the INM and lamins for proper mitotic regulation. Bulk of lamin disassembly occurs after the NPCs are pre-embellished. As the lamina is disrupted, many INM proteins lose their anchor and release into the endoplasmic reticulum. NPC disassembly is critical for early stages of nuclear envelope breakdown. Next one is the nucleoplasm. Nucleoplasm is a gelatinous substance within the nuclear envelope and also called karyoplasm. This semi-aqueous material is similar to the cytoplasm and is composed mainly of water with dissolved salts, enzymes, and organic molecules suspended within. The nucleolus and chromosomes are surrounded by nucleoplasm, which functions to cushion and protect the contents of the nucleus. Nucleoplasm also supports the nucleus by helping to maintain its shape. Additionally, nucleoplasm provides a medium by which materials such as enzymes and nucleotides, DNA, RNA subunits can be transported throughout nucleus. Substances are exchanged between the cytoplasm and nucleoplasm through nuclear pores. DNA replication transcription occurs peripherally nucleoplasm. Next one is the nucleolus. Most eukaryotic cells contain one or more prominent regions within the nucleus called nucleoli. The nucleolus was first described by Francia in 1781. Nucleoli vary in size in different cells. It is now known that nucleoli are the sites of transcription of the ribosomal ribonucleic acid genes, multiple random copies of the sequences encoding 3 or 4 RNA species present in ribosomes, and of the biosynthesis of the large and small peri-ribosomal subunits. These multiple copies of the rRNA genes are present at one or more chromosomal sites called nucleolar organizer regions. The NORs are the sites on the metaphase chromosomes, and there is a tendency for the NORs to close to telomeres on the chromosome arms.
The next one is the rDNA organization. In prokaryotes, such as Escherichia coli, there are three rRNAs, cystines, 23S, and 5S, which are organized as a single transcription unit. In all eukaryotes, the organization of the rRNA gene is recognizably similar to that prokaryotes, but with major differences. The size of small subunits RNA has increased from 16S to 18S, and that of the large subunit has increased from 23S to 28S. A new small 5.8S rRNA has become interspersed between the sRNAs and the iRNA, and the 5S rRNA has become separated from the other RNAs in a different transcription unit. The former transcription unit is generally simply referred to as the rRNA gene or the ribosomal deoxyribonucleic acid. The separation of the 5S gene is intriguing and so far unexplained. In most eukaryotes, the 5S gene have become completely separated from the rDNA's repeats at entirely different chromosomal sites and are located within the nucleoplasm rather than the nucleolus. 5S genes are transcribed by a different RNA polymerase from rRNA genes. The resulting 5S RNA molecules must be imported into the nucleolus, and the transcription must be coordinated with the transcription of the rDNA. Structure of nucleolus and rDNA transcription. Miller's threads. Actively transcribing rRNA genes were first visualized by Miller and colleagues in the 1960s. They lysed cells with simple detergent treatment and spread out active rDNA onto electron microscope grids. In images, line of a single DNA molecule can be clearly seen, with many polymerase molecules attached to successive transcribed regions. Nascent RNA strands can be seen emanating from each polymerase, the length of the strands increasing for polymerases further along the gene. Often knobs are seen at the end of strands corresponding to the binding of proteins and the formation of folded pre-rRNP particles. Because of their characteristic appearances, these rDNA transcription complexes have been called Christmas trees. Despite these beautiful and readily interpretable images, it is only recently that its view of the rDNA transcription complexes have begun to the reconciled with the image of nucleoli obtained by thin section electron microscopy EM of intact cells. Next one is nucleolar proteins. The nucleolus contain mainly different proteins, many of which are also present in other cellular locations. They include the ribosomal proteins, the proteins involved in transcription of pre-rRNA and its subsequent processing, and many other enzymes such as methylase, topoisomers, nucleases, kinases, and phosphatases. rDNA transcription is catalyzed by RNA polymerase, a complex of approximately a dozen protein subunits. Initiation of transcription requires the coordinated activity of a series of transcriptional activators and binding factors. Two of the most abundant nuclear proteins are nucleolin and fibrillary. Nucleolus. Contained within the nucleus is a dense, membraneless structure composed of RNA and proteins called the nucleolus. Same of the eukaryotic organisms have a nucleus that contains up to 4 nucleoli. The nucleolus contains nucleolar organizers which are parts of chromosomes with the genes for ribosome synthesis on them. The nucleolus helps to synthesize ribosomes by transcribing and assembling ribosomal RNA subunits. These subunits join together to form a ribosome during protein synthesis. The nucleolus disappears when a cell undergoes division and is reformed after the completion of cell division. The next one is chromosomes. The nucleus is organelle that houses chromosomes. Chromosomes consist DNA which contains hereditary information and instructions for cell growth, development and reproduction. Chromosomes are present in the form of strings of DNA and histones called chromatin. When a cell is resting, that is not dividing, the chromosomes are organized into long entangled structures called chromatin. The chromatin is further classified into heterochromatin and euchromatin based on the function. The former type is highly condensed, transcriptionally inactive form, mostly present adjacent to the nuclear membrane. On the other hand, euchromatin is a delicate, less condensed organization of chromatin which is abundantly in a transcribing cell. Besides nucleolus, the nucleus contains a number of other non-membrane delineated bodies. They include causal bodies, germany of coil bodies, polymorphic interphase, karyosome association PIKA, promyelocytic leukemia PML bodies, and paraspecicles and splicing specicles. Chromatin. Chromatin is a complex DNA and protein found in eukaryotic cells. Its primary function is packaging long DNA molecules into more compact, dense structures. This prevents the strand from becoming tangled and also plays important roles in reinforcing the DNA during cell division, preventing DNA damage and regulating gene expression and DNA replication. During mitosis and meiosis, chromatin facilitates proper segregation of the chromosomes in anaphase. The characteristic shapes of chromosomes visible during this stage are the result of DNA being coiled into highly condensed chromatin proteins. Histones and non-histones are two major types of proteins associated with DNA in chromatin. Both types of proteins play an important role in determining the physical structure of chromosomes.
Next one is histones. The histones are most abundant proteins in chromatin. They are small basic proteins with a net positive charge that facilitates their binding to the negatively charged DNA. Five types DNA: H1, H2A, H2B, H3 and H4. Amino acid sequences of histones H2A, H2B, H3 and H4 are highly conserved, evolutionarily speaking, even between distantly related species. Evolutionary conservation of these sequences is a strong indicator that histones perform the same basic role in organizing the DNA in the chromosomes of all eukaryotes. Histones play a crucial role in chromatin package. Importance of histone: N-terminals, tails. Histone N-terminal tails are central to the process that modulate nucleosome structure and function. The N-terminal tails of the four core histones are targets for post-transitional modifications such as acetylation, methylation, and phosphorylation that correlate with the changes in gene activity. Contribution of histones tails to nucleosome structure and nucleosome arrays has been demonstrated by several researchers, but the mechanism involved in acetylation mediated transcriptional regulation remain to be elucidated. Removal of the H3 and H4 N-terminal tails as well as the acetylation provokes a dramatic change in the linking number difference of the H3H4 two tetrameric containing tailless or hyperacetylated histones shows a striking preferences for relaxed DNA over negatively supercoiled DNA. Next one, non-histones. Non-histones are all the proteins associated with DNA apart from the histones. Non-histones are far less abundant than histones. Many non-histones are acidic proteins, proteins with a net negative charge. Non-histones include proteins that play a role in the process of DNA replication, DNA repair, transcription, and recombination. Each eukaryotic cell has many different non-histones in nucleus. In contrast to the histones, the non-histone proteins differ markedly in number and type from cell type to cell type within an organism at different times in the same cell type and from organism to organism. Next one is euchromatin and heterochromatin. The degree of DNA packaging changes throughout cell cycle. Two forms of chromatin are defined, each on the basis of chromosome staining properties. First one is euchromatin. It is the chromosomes or region of chromosomes that show the normal cycle of chromosome condensation in the cell cycle. Visually, euchromatin undergoes a change in intensity of staining ranging from the darkest in the middle of mitosis to the lightest in the S-phase metaphase. Most of the genome of an active cell is in the form of euchromatin. Typically, first one, euchromatic DNA is actively transcribed, meaning that the genes within it can be expressed. And second one, euchromatin is devoid of repetitive sequences. And the next one is heterochromatic. This is the chromosome or chromosomal region that usually remain condensed, more darkly staining than euchromatin throughout the cell cycle, even in interphase. Heterochromatic DNA are usually transcriptionally inactive. Two types of heterochromatin is present. First one is the constitutive and second one is facultative. Constitutive is present in all cells at identical positions on both homologous chromosomes of a pair. This forms of
Heterochromatin consists mostly of repetitive DNA and is exemplified by centrosomes and telomeres. And the second one is the facultative. Facultative is the, by contrast, varieties in state in different cell types and at different developmental stages, or sometimes from the homologous chromosomes to another. This form of heterochromatin represents the condensed and therefore inactivated segments of euchromatin. The bar body and inactivated X chromosome in somatic cells of XX mammalian females is an example of facultative heterochromatin. The next one is the telomeres. A telomere is the end of a chromosome. Telomeres are made of repetitive sequences of non-coding DNA that protect the chromosome from damage. Each time a cell divides, the telomeres become short that the cell can no longer divide. Telomeres is that in cancer cells which have a more infinite capacity for cell division. One of the most important changes that they make is that they keep the telomeres long so that molecular clock goes away and those cells can keep dividing even though they should get to the end of their lifespan. Conclusion. In above section describes that eukaryotic nucleus structure and its functions. Structure and organization of nucleus, nuclear pore complex, nucleoplasm, nucleolus, RDNA transcription, nucleolar proteins, chromosomes, chromatin, histones are described above. Thank you.